So I'm going to talk about something that's really very, very different. And I hope it's not so different that people aren't interested. But I hope to keep you along. And I think perhaps the main aim for me is, aside from highlighting some of the work that we do, is to perhaps, or what might be useful for you perhaps, is if anyone's thinking about data problems, particularly in the biological or medical biospace, that they're thinking of trying to think of a problem or something to address. Maybe there might be something in here that might give you an idea, or perhaps give you an idea of some of the problems that we're trying to address. And it'd be cool if anyone could do it better. We're not doing it very well, but we're starting to learn how to do it. So, I mean, I think there's probably lots of good ways. So some of the work I'm going to speak to is sort of a real good big collaboration between these four people. And so I'm representing these four people here today, really. This is Alexis Aiken, who's a professor in maths at UCL. This is Oleg Glius, who's a senior lecturer at QMUL. And this is Tatiana Nazarenko, who used to work with the three of us in Alexis' group. And actually, she did a lot of the sort of hard work, I suppose, with this, to get the nicer job of oversight. So I will try not to give you a biology lecture, but I do want to bring people along with me. So forgive me if some of this is a bit patronizing, but what I'm hoping to sort of persuade you of is why sort of our approach has some good rationale behind it. So I think most people will be aware of that we all have DNA in us. And hopefully many of you will remember from school that DNA has to act through an intermediary, which is mRNA. And we call that process transcription. And I will say transcription and translation, but I'll mean translation when I say transcription, and I do that all the time. But if you remember this slide, then it's fine. And RNA is not sort of the end product of this central dogma, but proteins are. And they're the sort of functional phenotype. So we make the proteins go into the cells or our tissue, and they do stuff. And that's what helps to make us when we develop and grow, and us when we have some beer and we need to get rid of the alcohol. So at the end of this, we have proteins, and they're doing a lot of the hard work. And obviously, this is not the full story, because this can't be going on for all genes at all times. Otherwise, we would just be a homogeneous blob. No comments on me. But so this beginning of this is the DNA expression of genes. So if we consider DNA is made up of lots and lots of units that we would call a gene, and most of those genes encode for a protein. The expression of those genes, some of the regulation of that is inherited from birth, or even before birth, so in the gametes. And some of that is sort of dynamic. And actually, it's a more recent findings, the last 20 years, that really realized that a lot of those regulations are more dynamic than what was previously thought. So this is both an inherited and a dynamic process, that actual gene expression as a function, the feature of the gene, so chemical modifications of the actual DNA can affect gene expression. But then proteins can also feed back to regulate exactly which genes want to be expressed at what moment. And that's important because we're going to have a beer. We don't want all our enzymes or all the proteins expressed that are going to degrade the beer all the time. We just want it to have it when we need it. And there's different ways that can be done, and I've just listed them, but I'm not really going to go through them. And the same for translation. So RNA has to be used as a template to make proteins. And again, there's proteins involved in that regulation. And actually, there's also RNA itself. So RNAs are quite a very diverse group of molecules. They're not quite just a single strand that is involved in translation. They have a lot of function as well. And sometimes they're interacting with proteins. So there's quite a dynamic interplay between a lot of RNAs and proteins that also regulate their expression. And then the proteins themselves, they get turned over, they get modified, they get activated, they can be moved across the cell, they can be moved out of cells into tissues, etc. Otherwise, obviously some proteins are secreted because that makes our hair or our skin. So they're not all just cellular and occupying in an aqueous space. And all of that has to be regulated so you don't have hairy lungs. So this, so it gets more complicated. The proteins themselves, they're all doing everything in the cell from this perspective. And here then is an example. So this is taken from Reactome, which is a database of protein pathways. And this is trying to summarize a lot of everything that the protein's doing in a single slide. You can go on reactome.org and have a look. It's a very cool and interactive website. You can look at all different pathways. And so this is sort of summarizing a lot of major pathways. And I've just circled one to sort of zoom in on. And if the resolution comes back, you might see some more detail. And you can see that within that sort of little group, there's obviously a lot more complicated pathways and things going on. And if we zoom in again, you can see that all these sort of individual nodes or bits of this network are interconnected. So where we have in blue, which you can't really read, I'll zoom in on the next slide, might be better. Okay. So you can see in blue we have these would be proteins here. And you can see this one, alcohol dehydrogenase complex. So the complex implies it's not just one protein, it's actually made up of more than one protein, which is another level of that sort of regulation. So protein is not just active on its own, it needs other proteins there to be active, and they're independently regulated, or they might be independently regulated. And also they might need other small molecules, or in this case, zinc to work. So this is a protein dimer that also complexes with zinc. And what you can see in green is that there's lots of other small molecules throughout the body that we also need. And they're not part of that central dogma, but they're obviously essential because we don't just eat proteins, we eat sugars and we eat fats. And all of those things are regulated and metabolized to make us and do everything. And all of these work together in this sort of network. And it's this network sort of bit that I want to focus on. And to sort of understand this network, we have to think of, I think of this as sort of a multi-layer. So you have genes occupying one sort of layer of a network with a multi-layered network where you have RNA and then proteins as well. And these pathways that we just showed maybe intersect that network because the proteins are regulated, the expression of the proteins are regulated from each of those nodes in the network. And all the proteins themselves are all working together to perform one task or essentially transfer one molecule down a chain. And we can analyze all of that. And this is what sort of omics analysis is. So we have proteomics and genomics. Genomics people may have heard of, proteomics maybe not, metabolomics. And each of these deal with a suite of analytical techniques for looking at that point of regulation or that point of data. So you have epigenetics, which are looking at the points of DNA or the modifications on DNA and the DNA structures that are inherited that can, or acquired during life, but that regulate gene expression. Genomics is looking perhaps specifically at the genome sequence and what's different between people. Transcriptomics is obviously looking at, well, not obviously, transcriptomics is looking at the transcript, which is the mRNA. So what mRNAs are present in the cell. And proteomics is looking at the proteins, and metabolomics is looking at everything else, all the small molecules. And all of these are kind of independent tools. But really what we want to do is understand all of this together, but mixing all of that data from independent analytical tools. And we're talking about physically different here. So not conceptually slightly, but also physically different. Is quite challenging because each of those methods has their own constraints. So how the data is generated and what the quantification means and what the word's gone. Anyway, you get the point. So all of those are independent. And part of the problems, okay, so what is the point? And you'll be relieved that that's more or less the end of the biology lecture. My point is that biology can really be considered as a collection of networks. What I've just shown is sort of molecular networks, but you could expand that. You know, we have organs, we have cells and organs and tissues and organs, bodies, different people. So that network can expand. And really what I want to do is study these networks, or these networks themselves are kind of studied using discrete techniques. And each of those techniques generates tons of data. So my speciality is proteomics. My bread and butter is not data science, but I've come into it, so you'll see where it is. My bread and butter is analyzing proteins by mass spectrometry. So I can take some tissue and in about a day of messing about in the lab and then 20 minutes on an instrument, we can analyze maybe 7,000 to 8,000 proteins from that tissue and tell you quite a lot about them. And that's just one of these points on this network. So we can get a lot of data from one person. And obviously, if we're analyzing a cohort of people, that's reproduced across potentially a large cohort. And the pathways then intersect the network, and want to understand all the points of those intersections to understand how all these proteins and how biology is regulated and how that relates to disease. And so sort of the ambitions in my group is really to understand the networks and how they relate to biology, combine some of these discrete techniques into something that produces sensible data sets. And then what I'm really going to talk about now is identifying sort of unhealthy features in that network. So can we generate a network and predict something about disease, whether or not you have a certain disease or what your outcome from a disease will be, which is sort of a point of them both. So this is the problem that that brought me into this field. So we had a study on, this is a few years ago, looking at ovarian cancer. And ovarian cancer is quite well-known marker, which is CA125. It's used clinically. You have to have other symptoms, and if you have other symptoms, then they may test for CA125 to see it's elevated. There are other markers that throughout the literature and other research are also quite well known, at least to be okay biomarkers. They're not quite good enough to be clinical. They don't beat CA125, but potentially in combination. So we thought, okay, we have quite a small study group here. We'll analyze a select few proteins. So we're not going to look at thousands. We're going to look at what we think might be kind of relevant, and then see, okay, can we build some better models to predict? And this is a problem, or one of the starts of the problem, is that when we actually just first had a quick look at the data, we see CA125 is up. Nothing else was really changing between the two cohorts, at least on their own. Despite the fact that we know this HE4 that I've highlighted isn't important, and we expected it to be changing, because we know that this is a subset of another data set, and we know overall in this data set that HE4 actually changes quite a lot. So maybe we were unlucky, or maybe the larger cohort was lucky. But anyway, it didn't change in ours. And so what we actually then did with the study is that we thought, well, actually just looking at an individual biomarker and say, okay, if it's above this threshold, if this protein or molecule is above this threshold, we're going to say you have a disease. That's not very personal, and it's not very good. Normally, proteins or molecules are probably expressed something like this. Some people are just naturally high, some people are just naturally low. So what actually is better to look at is how that marker changes over time. Which is what it's going to work. So is what this is showing. So we have healthy people here, and you can see already, you can see that there's a range. So this is the CA125 marker. You can see there's a large range in the healthy cohort. So already some of those are naturally high. And as the disease progresses, which is this orange cohort, you can see that this biomarker jumps up. Whereas in the healthy controls, then actually nothing is changing over the same sort of period of time. So what's actually important? Yes?
Isn't it too late when it's jumps up like this?
Well, you'll see in a moment. Not necessarily. I mean, a good biomarker should tell you early enough to treat the disease. Otherwise, it's not such a useful biomarker. Unless you're sort of a pathologist, I suppose. But that is too late, right? Right at the end, I'll show you some Alzheimer's disease data. They were all dead, because that was from the brain. So there's this change over time. And we can measure that, because if you have a screening program, you can measure something over time, and then you know for a person if that is changing, and the rest of the population is sort of irrelevant. As long as it's changing in you and you can measure that, then that's useful. And so we can do that. We can generate a model that has different proteins in it, and we can improve prediction slightly over just looking at CA125. So that's nice if you can do something screening. Look. And it generates a personal test, because it's about you, not you in relation to a population. But screening is expensive, and it's not always an option. There was a huge screening study, which many people probably don't know, but it was in the news a few years ago, called UKC Tox for ovarian cancer. It was huge. 400,000 women monitored over 10 years. So millions of samples. And I think it still is the most expensive and the largest, UK at least, screening study. And the result of it was that they could identify ovarian cancer earlier, but there was no benefit to mortality, because you're still not quite identifying it soon enough. The quality of treatment is not good, and the cohort is also aging. So if you're only identifying it in much older people, the chances of survival over 5-10 years, which is sort of the relevant clinical sort of milestones, are already sort of capped, because they're already older and much more likely to have other comorbidities. So screening is not always an option, and it's not always the best solution. So we wanted then to take that data, so can we look at it in another way? So not just looking at individual markers or trying to pull out combinations of markers that do well. Can we look in an individual at all of their proteins all together without overfitting, and find anything useful for prediction in this manner? So we construct this sort of network map. And the whole point of the first half was try to persuade you that looking at these in a network context is sensible biologically. Although there's no biological information, I'll describe how we make these networks, there's no biological information in the network other than what the proteins are, which is slightly different to the network. So what we generated these networks, and this network shows for an individual with ovarian cancer what is changing in that individual compared to a population of healthy people. And what we can see in the network is that the really important, I just flick that back up for memory, is that these really important proteins that we know are changing are really important to the sort of the main structures within the network. And these are the two that I mentioned, but there are others in these grey and blue clusters as well that we know from the literature are really important. And those are central to the sort of shapes and topologies in the networks. So what we do in this case is we wanted to see, okay, can we identify any common shapes, features, topologies, we call them, of the network that we can use to predict the disease. And so this creates a challenge. How do we actually make the network? Because I said we're not putting any biological information. So what do those links actually, connections mean? How do we make a network when one doesn't immediately exist? And kind of mentioned these genetic, epigenetic, proteomic, and these pathways, that these networks that I've described that exist in biology to begin with. And you could say, well, we can identify the proteins and we could do some gene sequencing. So we could actually infer some biological networks for these people and try and use those. But there are kind of problems with each of these. The genetic epigenetic is quite static. Epigenetic a bit less, but it's still reasonably static. So if you have cancer where you've developed new mutations, or maybe not cancer, but if you have some other disease, then actually your genes aren't really going to change. At least the gene sequence is not going to change. In cancer, you may get some very small localized mutations that lead to changes in genes, which is what's important in the onset of cancer. But you've got to be able to find that cell that contains that change. So actually, in a practical sense, for predicting at birth what somebody might be more susceptible for, useful, but for predicting a disease they may acquire or at what point they're going to acquire it later in life in a more dynamic context, not as useful. We may think, I sort of mentioned that there's all these biological pathways, that proteins are all involved in pathways, but a lot of proteins we don't know all of the pathways are involved in. So if we apply that biological information onto a network, we may end up going down a bit of a rabbit hole, or we may be missing something that's kind of important. So it can be useful, and in fact, it is very useful to map straight onto pathways. But in a biomarker context, when we're trying to discover something new, it can be a little bit limiting. So this leaves us with proteins, and my background is proteins, and I like proteins because I'm a proteomic scientist, so this is all very nice for me. And I'm not going to talk too much about this. This is sort of some stuff from another talk. So how do we generate these networks? The networks themselves, so this is really just saying that it's an evolution as to how we've generated these networks. It's not just sort of came up with it overnight. They were described in 2014 as one way to generate them by a different group. And there are other ways of inferring networks. There's some reasons why we like this way. And so we made a big conceptual change to the networks, and then we sort of developed them further on as well in these 3 papers. So how do we make them? And I think this highlights another sort of key challenge is not very scalable, at least for us anyway, who don't have your GPU, but maybe we need it. So our solution, I'll tell you what our solution was in a moment when I describe the problem. So this was fine if you've got that data set, which was about 120 proteins, do it on a laptop, it's no problem. What we do is we take, you go on 2 clickies. We take a population that's got disease or not disease. And we then generate, and we pick any 2 proteins, in this case, from that population, any 2. And then we generate a model within that. So it could be an SVM in this case, a support vector machine. And that just produces a probability map. What's the probability of that pair of proteins coming from somebody with a disease or without the disease? And then we do that for every single possible combination or pair of proteins. So if it's 120, it's not so bad. The data sets we've done for Alzheimer's, this is 5,000 or over 5,000 proteins for hundreds of people. So that becomes a big scalable problem because we've got millions of pairs over, and you've got to do it again 400 times. And if you do prediction, then you might want to do some cross-validation and things like that. So that becomes a big scalable. Our solution for that was to get a master's student to generate, to just break it down into sort of chunks of 100,000 or so and find as many laptops as they could. So this is a network, I think, in fact. But that worked. We got all that data into it. But that is another story. So what we then do, so if we do this for every single pair, we then take an individual who we don't know anything about. We take the same pair of proteins and we say, okay, what's the probability of that pair of proteins belonging to a case or control based on those previous models? And then we repeat that for every pair of networks so that you get a network for that individual that represents how all of their proteins are different or are likely to be disease from a disease or not. And then we take that network and we look for shapes that are common between people that we know the eventual outcome for. And there's some really good benefits for doing this that we found. So all those shapes you can define in mathematical terms, and then we use those definitions to generate a predictive model. And we see quite clearly that if you just train a model using whatever you want, neural networks, this is an elastic or a SU restricted general linearized model or random forest, you can get much better improvement if you use the topological features of the network to test those models or build and test those models on than you do just straight off the raw data. And some of that comes from the fact that you are reducing the data down to sort of fewer features. You might have less overfitting problems, but it also comes from some of the redundancy and properties of the network that really help benefit disease prediction. And some of that comes from the fact that they're less sensitive to noise. And if you think that we're identifying 7,000 proteins, most of those aren't going to be related to the disease. They're just going to be what your body's always using, or they might be related to something else. So in our context, they are just noise. But the networks are much more resistant to that noise is what we find on this data. So if we make this an artificial data set where we can control the level of noise, we actually find that we can still train and test and generate robust models using the network topology of features. But in some instances, in very sparse networks, it can help to have a little bit of noise because it helps define some of the shapes a bit better. And finally, for this omics type of data, so in omics, proteomics, we get like 7,000 proteins, maybe. If we have a small cohort, so normally we don't have 7,000 patients. We maybe have, in that study at the beginning, 30. In the Alzheimer's disease that I mentioned, we had about 400. But that's still a very small number of individuals compared to what we can measure in them, which results ultimately in an overfitting problem, a statistical overfitting problem. And actually, we find that with that type of data, again, using the topological features of the network are much more resistant to that overfitting. So these networks have been used in quite a lot of different ways, not just by us. So this is the group that actually first described them in 2014. And they described them at that time for a slightly different application than prediction. We sort of, our group, the main thing that we've done is develop them as a useful tool for prediction. And they've applied it to things like credit card fraud. So a group in collaboration with Tatiana and Alexei used these to look at DNA methylation changes in Down syndrome. I can't quite. Our ovarian cancer study, when we applied this approach, we found we were able to diagnose 3 years earlier quite a large number of patients that weren't identified. So that kind of brings on to your point, I guess. Is that biomarker any good? This one's better. And then in COVID, we also were involved in a study with a German group where we were able to use these networks to predict outcome with an unerring accuracy, which we were doing more or less live. The hope, obviously, being that the doctors in intensive care could change the regime. So we know that for prediction, they're really good, but also we're not just interested in prediction. I want to come back to those earlier problems where we want to use those to, can we actually infer any useful biology from them? And so I'm going to use an example. I'm going to be quick here, but I'll use an example from the COVID paper. I'm going to skip some because you won't be too interested in the details of the paper. Okay, so here's 2 examples of networks, of the actual sort of real networks from the COVID data, and the probability is the probability of survival, or no, the probability of death, sorry, from being on intensive care. So you can see that this person had a very high probability of not surviving on intensive care. This was about 30 days before they actually passed. And this person had a high probability of surviving, or a low probability of not surviving. And you can see visually that there are differences in the networks. And these are quite representative examples. So, okay, so are those differences actually useful? Do they tell us anything biologically? Here is just another convincer of that. So you can also classify somebody with COVID as to how severe that disease was. The World Health Organization quite quickly released a classification system. If it was ordinal, you would be scored 0 if you didn't have, 2 to 7 if you were on late stage intensive care. And you can see that progressing through these different scores, the networks also resemble quite different shapes and structures within there. And that's visual. And these are the sort of numerical descriptors of these shapes in there. So you can see that these correlate very highly with the change in COVID severity. Okay, so the actual original use of these networks was to find biological meaning, or try and find a new way of saying, okay, this gene is different in this. And this was done in some plants, Arabidopsis. Arabidopsis is a really common sort of biological plant model that geneticists often use. And it kind of worked. There were some big problems with how the networks were generated that we overcame in our data, but this was the original use. And I think in our ovarian cancer study, we can also get some hints that some of these proteins that were involved, particularly in these clusters here, actually could well have some biological meaning early on in the cancer progression. It's a bit harder to test that sort of thing. And in the ovarian cancer, if we generate these sort of average networks, if we take all of the networks and produce an intersection of those networks, so what is on average changing, then we see that the features or proteins that were most important to the shapes within that network were also important biologically. So of the 25 proteins with the highest relevance, 15 were part of the coagulation system, and if you kept up with the news, then blood clotting and things like that was an issue with COVID. And 8 proteins belonged to the complement cascade, which is related again to inflammation and the sort of skewing of information that was associated with COVID. So again, in this data, it looked quite like it. And so this is my teaser to sort of finish with, which is gradually getting more busy as a slide as we get further along this work. But we wanted to then say, okay, so we think from these network structures are really good for prediction. Are they also good for inferring biology? And so we've applied this to this Alzheimer's disease data set, which is a data set that we acquired using sort of in my lab with a collaboration with Joe Jackson from UK Dementia Research Institute. And what we find is that, so don't worry about what it says, but what these green dots here, these are all pathways that are identified from that data using quite conventional univariate or multivariate statistics that we would use routinely. So you can see we actually identify a lot. These two on the outside are pathways we can identify based on features that were important in the network. And what's kind of useful, interesting to us is that in many cases, these are completely distinct. Okay, I appreciate there's more here, but there's other reasons for that. But we have a good overlap in very highly probable and highly likely pathways. And then the others, we have completely distinct pathways that look relatively new in terms of the context of Alzheimer's disease. Here's another problem. One of the problems with Alzheimer's disease research is if you find a protein or pathway you think is interesting or important and you Google it, you will find there's so much research on it, you will find a paper that says, oh, it's important. So this is also quite hard to validate, and that's where we are at the moment with this. So we're sort of reproducing some of this data in some other data sets to see if we get concordance, and then we can try and develop some experiments to sort of test these out. So with that, I think I will end on a unpublished. So that comes with a caveat, it's not published. So I think one of the sort of ambitions is that, which is not just from me, there's lots of spin-out companies trying to do what I was about to describe, but it is a big problem, and there's no one doing it really well at the moment, although everybody says they can, is that can you take an individual, go and have something that's reasonably non-invasive, like a blood test? Can you generate some network that relates to the biology of that individual, regardless of what you're measuring, but can you generate that sort of network? Can you interpret that network to relate to some biology and then make predictions about what they're going to do? And if you can couple this sort of network approach with screening, then you have an extra sort of layer of personalization, and then you can monitor changes in lots of different things. And this is sort of a, big blue sky vision that we have in our lab. I don't think we'll ever get there or not from us, but there are lots of groups sort of working on this, and it is a big and interesting problem. And there are bigger and bigger data sets for people to go out and test and generate models that may be capable of doing this. In fact, the UKC tox data sets are all available, and they're also really useful. In fact, there's lots of COVID data sets. If people are interested in that sort of problem, there are data sets where you can go and have a go and see if you can develop something that's kind of multi-disease or fixes. So with that, I will end there. Thank you. These are sort of the people who are involved. So this is the top left is the ovarian cancer group. The dementia research data, which I showed right at the end, is the corner, and this is my research group. Shahid at UCL is continuing the work on the Alzheimer's disease. Sorry.
So two things, in silico versus in vitro. Comment on that. And the second is we heard 600 million sterling was spent on an Palantir project cancer. If you can comment
On a what project, sorry?
Palantir, the American software company that uses the NHS spent 600 million.
Oh, I don't know. Some of the details, though, but I don't know. So I can't comment. In silico or in vitro, I mean, the data sets are there for in silico use. And provided the data sets are well described, then there's lots that can be used for those. In terms of, do we have enough data to generate in silico models of these networks that really relate well to disease biology? So can we take some data? Okay, so we know that this protein interacts with this proteins with these metabolites. There are groups trying to do this, and they tend to do it in very closed systems. The problem is it's twofold. One is computational, because there's a lot of different. At some point, you've got to go from adding in degrees of uncertainty into those models, which come from many points, to how big you extrapolate those networks for them to actually be relevant biologically. So you could say, okay, let's consider all proteins involved in these pathways because they're involved in the disease we want. And there might be some data on those pathways that you can generate a silico model for it. The limitation there is that a lot of the proteins are also going to be involved in something else. And so if they're doing that, that will impact on the in silico model that you've done. You may also have proteins that you don't know about and how they impact on it. So I think that the groups that are doing it are generating quite limited data that in very narrow examples may validate in an in vitro situation. But when you try and build that into a sort of a larger population, including more variants that occurs biologically, I think they are not there yet.
This is a question that I asked Palantir with use of pattern recognition. So if you want to just comment on where in the slide you pattern recognition.
I mean, so we're not using any pattern recognition per se. We're using sort of mathematical descriptors of the network structures. Alexis Aiken is using, is trying to generate these networks for reducing dimensionality in images with the idea of using more pattern recognition. We also have a lot of pattern recognition in the context of imaging, which is another sort of field that we do, not light imaging, but mass spec imaging. So we can image all the molecules, if you like, in a pixel across a tissue and generate a map of those. And so there's a lot of challenges in the quantification resolution adjustment of that sort of data. There's some pattern recognition, but that's not really my field, so I don't think I can comment overly well.
I just had a general question. Just, it's well known that dogs, for example, can sniff out cancer. They can tell when a diabetic person's going to have a hypo or people about to have a fit and that sort of thing. And there's even the case, I think, of a lady who's able to predict Parkinson's, yeah, by as well. So when you're looking at your protein markers, is there anything on that field that you can use as a combination that's generating a particular smell that might be able to?
So Parkinson's was really interesting, and that's released in the sort of sebum in the oils on your skin. And the lady is doing a PhD in the group in Manchester. And they're able to, they have identified some of the markers that that person is smelling. In our context, I think there's not that many. I mean, these volatile solvents that people smell tend to be smaller molecules. So it's not directly my field. So in these networks, what the data we're using, no, not directly. But we are doing some groups where I work, they are doing things on breath. So we can breathe directly into the mass spectrometers. The mass spectrometer is a £1 instrument. You're never allowed to go up and breathe into it, but we can go and breathe into it, and you can see what's changing. So I'm asthmatic. They made me run. If you make me run up and down the stairs, you can see pyruvate and lactate changing differently to people who aren't asthmatic, or so they tell me anyway. In 1 versus 1. So that sort of research is interesting, and there is lots of other sort of sources. So breath, for example, that could be really routine. We're also, there's a group developing a toilet that can measure markers in your feces. So that is a real possibility, actually, that it's got some good traction behind it. There are some tools that are used in surgery that use mass spectrometry. So they thermally ablate the tissue, and molecules are detected in the mass spectrometer and tell a surgeon with a big green light, red light, if this is cancer tissue or not. And essentially the toilet works off the same kind of technology. So I think these sort of more ambient and really accessible approaches have got good traction and probably will get there. They are more newer technologies, so they are a bit behind, and there are quite a lot of distinct difficulties that people are overcoming. But I think they will get there.
Last question.
Quick question. Have you built a knowledge graph to identify your bandwidth?
We haven't. So we haven't built one. So we use the knowledge paths that are essentially online. So we use the Reactome data slide that I put up early on. They have a large data set of known pathway interactions. So that's what we mapped up. There are others, and they all have some limitations and things like that, as you would expect. But Reactome is quite good, and it's run by the European Bioinformatics Institute, which may actually be a useful talk here. They do a lot of modeling and a lot of large data curation and storage and annotation. That was the last question I was out of there.
Thank you for
Thank you very much.
Let's feel this was just really much the surface, and like if you have much more questions, just go talk to her in person, or I think you can reach him in LinkedIn. So, thank you. And we will have again a few minutes of a technical break, so if you need to loo or just relax yourself, take your time, and then Sethu will be talking about reference.
So you yes and no. So we're not directly, but again, collaborators, people who collaborate. No, so that project is fairly early stage. How we use them to predict things. If we're going to use topologies, we have to at some point apply pressure. With the graph neural network, it's certain methods. I think it will. I think the prediction is logical. Where the group more interested actually just my group more interested in pages. I think with the graph, what we like about them is interpretation. We know how those images are what they represent. Everything that
Are you LLMs? Like this is more
How are you?
Yeah, working on it.
I'm very interested in balance. That's why I want to talk to you. I've got the right
just based on the sequence. So we had some discussions recently. We have some
yeah, exactly.
similar sort of problems. So we had that same similar sort of we had a
you're allowed to How can you find it?
That's
But those people that they somebody else might, right? Not you guys. That's the main one.
I need to figure out like I need to figure out the notes. This one is I change the screen size, but then sometimes that's fine, just push Facebook. So I can just like hold on like this. But I want to see the notes though. I want to see the notes.
So it's used as a data set of known. So when people do you tend to see that.
I'm fine here. There's that way. But then it's right there, I can't see it. Mouse? I have it.
Whenever you go next slide, have a look.
That's a good idea. Yeah. Use phone to see the notes. Bro, that's good. What do you need? Look at it. Everyone's around there, bro. 2x. Oh, shit. Okay, hold on, bro. How do I answer? We need to find my story. Right now?
Everybody in the ring.
Okay, then,
Yeah.
I need to join this call, share my screen. Once I finish, I'll then share my screen and that's it. 7 January, hopefully it goes fast.
But these, I mean, these are
Yes.
It's fine, it's fine. You know about the atrocities?
it's a real problem. One of the problems is that if you don't have large
What do you think like that data analysis analytics?
why a lot of studies I want
All you need is AIP. Yeah, attention is all you need. AIP is all you need. It's gonna be. I'm gonna ask you. So now that we like actually like hit 8, do you know what time we should be like?
Yeah, so in the same, we should start. We are a bit late. Sorry,
So
you lost.
yeah, I have it already. So, do I have how many? Do you have actually 30 minutes?
Yes,
That's fine.
yes.
I'm gonna start putting my notes here. You can easily.
So, I mean, a lot of it is a problem, and a lot of reasons we study.
Yeah, Wi-Fi?
Yes. So
that's a bad thing. That's the question you wanted to maybe.
But that's not necessarily good for NHS.
The other interesting thing when they come over from another country,
environmental diseases and how you interact with the environment. So
We don't need to.
it's fine. I mean, I have.
Generally speaking, right now, sometimes we are, but generally you capture a certain snapshot of that person. Don't know history. You don't know their own name. So, yeah. I think that's, you know, so his analysis of the data
Why isn't it on here?
is allowing it. So I would say if you approve the 20 years, sorry,
I can't, David can let me see.
Yeah,
Just a
very
moment.
one snapshot.
I just want to get the note on board. No, bro, it's a lot of things. It's a lot of there's layers. And then to choose what the interaction.
Just a moment.
Yeah, no, it's if you don't.
Oh, there you go.
I also go instructive. Yeah, okay, it's good to send me like split screen or something.
I don't know whether you can.
I think it should be like that. Oh, you could just put it down here. And that's it.
There's a lot of like video talks about like training data. The data's just too complex to just.
It's a good idea. There are groups that are trying to acquire more and more data with a view to instruct like within up, but that sort of thing, potentially, you may be able to get.
Yeah, sorry.
You're always going to be a little bit knowing the limits of you do it. How do you find that person? He wasn't happy.
Cool.
Like that. It's the last slide.
Can I see the notes? I just do this.
You can do present.
No? Yes,
Yes. Yes. You will have the recording and we will share it with, and every time we show that we from a technical issue we did it.
Sorry.
Just wait a few minutes. He wants to. Yeah, just wait a minute. Let me send something.
Yeah, okay.
Especially when the train gets when you move. So this is on the train.
Cool, let's go. Yeah.
Thank you for your patience. Please, everyone, stand right. You keep set a little bit more up close, so if you're already confident. Thank you very much. Please start.
Yeah, cool. Good evening, everyone. I am Sethu. Today, I'm going to be talking about GraphRAG. Before we do any of that, let me just give you a quick fact, some interesting fact. So research shows that every person is connected. It has a 6 degree of separation from another person. I know this is quite controversial, but I'm just going to assume it is true. There has been some research. Anyway, the reason I'm telling you this is basically every person in this world is connected by something. So it's a relation, as like an inheritance, anything. There are many ways to connect each other. So I guess if we actually think about it, there's an entire field of this relations and people between relations and like nodes and stuff. This is basically called graph theory. So we're going to take small piece from that graph theory and we're going to mix AI with it. And people have already done it. It's actually GraphRAG from Microsoft. So I'm going to talk about that in just a bit. Cool. So I'm just going to go next slide. I'm actually operating from my phone. So, oh, there you go. Okay, a little bit about me. I was a software engineer until like 2 days ago. I left Microsoft. So when I was at Microsoft, I was my task was to basically fix production bugs. Some insane like bugs I come across are like half the country of Norway has reduced a voicemail service. Can we fix this? And then some of them are AT&T. So my job was basically around fixing production bugs so that Microsoft won't have to burn millions of money as a penalty. So I was there for 1 year, and prior to Microsoft, I was at uni where I did my thesis in AI for fashion. I say you can see the strike through there. That's actually the name of my dissertation. I know it's going to be really boring to just write it down, so I just wrote AI for fashion. Anyway, so what I did was I trained a GAN model. For those who don't know what GAN is just a type of model that criticizes itself and learns from it. So given a clothing item, black hoodie, and my actual body, the role of the network is to mix that. So can we generate a realistic realistic virtual Tron image. So that's my dissertation. I also had a little fun in the end with stable diffusion because that was cool at the time, and I fine-tuned a model to essentially like make clothing items on human bodies with text. So that was a fun little project. Thanks to Krishan there for the idea back in the day. Cool. Prior to that, I was a placement software engineer at STFC, UKRI. For those who don't know STFC, STFC is Science and Technology Facilities Council, and UKRI is United Kingdom Research and Innovation. Quite a mouthful, I know, but so that was the research company. So there was a particle accelerator, and there was some neutron experiments, neutron muon, and they get the data. They want to analyze the data. There's a software, but that software is old and written by scientists. Let's just keep it that way. So it's like no software like life cycle or anything like that. So my job was to like bring it up to speed, add some cool features. I converted that. I had a bridge that allows you to run C++ functions in MATLAB environment and vice versa. So very niche, no one cares about thing, but it's just cool, I think. Cool. Let's just go next slide. Cool. Yeah. So today's agenda is to basically provide intuition of GraphRAG. I'm going to talk about what it is and why is it better than standard or naive RAG, and how does GraphRAG even work, and limitations of GraphRAG, and considerations for GraphRAG if you want to use it in your production environment or whatever. And there's going to be a cool demo where I'm going to show you the graph and infer hopefully from questions from some of the audience here. So I'll also, if you have questions, I'll take it in the end. Just I'd say, cool. So what is GraphRAG? So GraphRAG is a structured hierarchical approach to retrieval augmented generation, RAG. So it leverages the power of knowledge graphs to essentially answer user questions. So think of it like this. You have, just before I do all of this, how many of you actually worked with RAG? Raise of hands. Oh, that's a lot. That's pretty good. How many of you have worked with GraphRAG? Oh wow, this is going to be a learning experience, I guess. How many of you heard of knowledge graphs? Okay, that's good. Cool. So anyway, so knowledge graphs are the basis for GraphRAG. So knowledge graph means, let's say I'm a person, so I'm a node. You're a person, you're a node. The relation between you and me is an edge. Node, relationships, edges. Nodes, entities, relationships are edges in the language. So the actual power of GraphRAG comes from building these relationships, building the knowledge graph, and then being able to query based on that grounded knowledge graph. So that's the idea of GraphRAG. So instead of using a vector database, as naive RAG, we will use a graph, LLM generated graph, and then query from that. So that's just a basic high level overview.
I wouldn't agree with classifying vector database or search engines as naive instances. They are good in really search analytics. They are really good in textual search. Graphs are not great with textual analysis.
That is a great point, and that is why there is two methods in here. There is one graph search, there is an actual vector search. We actually don't want to forget vector search. Vector search is still actually really good and solves a lot of problems without having to rely on computational intensive graphs. And in the coming slides, you will actually see this. Microsoft GraphRAG will actually use both of them. So yeah. Yeah, cool. Let's go to the next slide. And yeah, so what is GraphRAG actually good for? So why do you want to use GraphRAG instead of RAG? What's the use case there? The use case is if you want to find recurring themes or like information about your data that will only be available to you if you have comprehensive understanding of the entire data. So let's say if you ask, you have data of like IMDb movies. Let's say like what happens in Batman, like you ask a question. Sure, vector search, Batman, like the query has Batman, you find the Batman in the data in the vector database, you can get the answer pretty easily. However, if you like ask question like how many movies have villains where villain is one-eyed? How can normal RAG even like understand what's the question? You have one eye, you have villain, like it's just not feasible. And even if it returns 1 or 2 results based on the vector search, it's not going to be a comprehensive answer. So that's where the GraphRAG really comes through. Because when you index this information of IMDb movies, you will actually create villains as a node, one-eyed villains as a node, and then link actual villains to that one-eyed villains. So it's all about like being explicit in your spreading of data and then being able to use that spreaded data. Cool. Yeah, obviously different perspectives, political opinions, like you can like summarize them, and there's also bigger picture. So like I'm gonna get to that in just a bit. Ah, there you go. Latency. Cool. So bigger picture. So what I mean by that. So let's just take an example here. This is not an actual like representative. This is not like image you want to give to your RAG. This is just an example of what naive RAG and GraphRAG looks like. So naive RAG, let's say you give a picture, ask the question, what are the people in the village doing? So it will look at people, village doing. It will find in the data set actions of people, village. So like it doesn't actually look for like in a specific way. It just like does vector similarity search. And then so in the actual information, let's just say the information is not an image, but a boy is pointing at the sky. A boy is looking at the sky. A gentleman is looking at the sky. So let's just say it's like individual piece of information. So when you ask, the answer would be a kid is looking at the sky. Another kid is pointing at the sky, and calf is drinking milk and behind. So that's just like completely separate answers you get. So that's just like not useful. So instead, if you ask GraphRAG, it's gonna use these individual informations. So each information is a node, and there are nodes that combine this information and then combine them. So it's like a tree that converges to one node at the top, which contains the entire image. The entire image, if you summarize everything, it will say there are planes flying, people are watching down from the below. So you will get the hard answer easily because it's the closest to you from the top. So anyway, that's just like a quick small explanation. Let's go to the next slide. Cool. So I'm gonna explain you what GraphRAG actually contains. So there's just like any other RAG system. There's 2 things: indexing, querying. You index your data once, query it forever if you wish, or until you got new data to index. So indexing is a 4-step process. So I'm going to talk about the steps just a bit, but it's a process in which you generate the knowledge graph using LLM. So that's the interesting part. And then because if you think about it, generating a knowledge graph is not exactly like it's not easy, because you have to first, like for example, I'll give you an example, right? John has a phone. Okay, if you want to convert that to GraphRAG, you identify what are the things involved. John, a person, node. Phone, a person, an object, node. And then owns, relation. So you have to be able to put your mind into it and extract this named entities and then relations between them. And imagine doing that for entire like BBC News over the last 3 months. How long is it going to take you? Exactly. So that's where LLMs really like come through. So yeah, so that's indexing part. I'll get to it. Querying. So answering users' questions using knowledge graph. This is pretty simple. You use the knowledge graph to answer questions. And there are 2 types of searches here. Searches as in the way you answer. So one is global search. So global search means it uses entirety of knowledge graph to answer your question. Local search means it uses specific things. Let's say you ask specifically about AI super cloud clusters. It will use that node and then spread around it, gain the information, answer you. So yeah, so there's 2 types. And some examples are what are the 5 most important events to review in the dataset. That's like a global question. And a local search question would be, what is Disney's lawsuit about? So like just like a little bit examples. Cool. Let's talk about indexing. Indexing is the process in which your private data becomes a knowledge graph. This is probably the most important one. So there's 4 steps: chunking, extraction, community detection, and community summaries. So chunking is the process where you take a document, break it down into manageable pieces. Cool, easy enough. You got extraction. Extraction is what I told you earlier. Like, John has a phone, like, the extra process of extracting entities and relationship between them. So that's the second step. And then third step, community detection. So that's going to be, that's an interesting one. So let's say John has a phone. Yeah. There's another sentence. John's brother has a phone. John's brother has a red phone. John's brother has a green phone. So let's just say there are some random sentences we found. If you want to like make a community, what I mean by community is phone, phones. Like red phone, green phone, still a phone. It's a part of phone community, right? And that's a level 1. And level 0. Level 1 would be colored phones, non-colored phones. So this is abstractions basically. It's sort of like hierarchy. So that is what community is. So you detect communities through LLM. Take this, find me the communities. So you build your graph from bottom up. You start with entities, find level 0 like community, and then use those communities to make even more abstract, like high-level communities. You keep doing that until there's nothing left to like summarize and like make another node of. So that's community detection. The way it happens is you use something called a Leiden technique. So all it means is it's basically really good at grouping things together, and it does this recursively. Like I said, you start from bottom, make some communities, do the same thing. That's the recursion. So every time you recurse, there's a level number. So level increases. Level is really important because that is what exactly you use when you're querying your global search. So if you want to search something in the graph, using that, if you want to global search something in the graph, you specify the level number along with your query, so that you will start at a point where you can use. I'll get into the global search later, but it's an important thing. That's what I'm saying. Community summaries. So cool. We got some entities. We got some communities. We got some even more abstract and high-level communities. How does the LLM make use of this data? There's so much data to work from, like so much data to look into. If I go if I take one node, go to left leaf and just keep going down, it can go forever. Like how does the LLM like even figure out what to look at, what not to look at? So this is where the map reduce thing comes from. So let's just call it process of elimination. Every time you make a community, there's a community report. Community report, it says what is happening under the community. How many nodes are there? What are the relationships? It's a report of that. It's a full-blown report. Then you make a summary of that report, so that when you query, you use the summaries to identify what is relevant for the query the user has asked, and worry about those summaries and leave the rest. Process of elimination, reducing your search space so that you have right things to focus on and important things to like consider when you make the final answer. So this is the visual representation. Documents to text units, chunking, text units to graph extraction, and then extraction to like summarization. So like this is like visual representation. Cool. Now let's talk about local search. I mentioned previously there's 2 types of searches. One is global, another is local. So local search is best suited for questions on specific things with less number of relations around it. So think of a big graph. You ask a specific question about like, I don't know, like a laptop. So you go to the laptop node. Immediately you can go there because you make embeddings for everything. Every entity has an embedding. So when you have embeddings, you can easily like do a similarity search, find the one that you need to focus on, go quickly there. So that's what local search is. It uses vector database like semantic search in the context of knowledge graphs. So the way it works is it's pretty like similar to vector search. So you get the semantically similar, like let's say like small example, right? I want to figure out who John's brother is. Let's say for example. And when I say John's brother, John is in there, brother in there. Brother, probably there's no node. Maybe there is a node, but it's probably not like, like it gets eliminated. So John, so we immediately jump to John node and see relations around John. Where John has a brother, John has a phone, John has a brother that is important. So I'm going to consider that. So you gain this information. So you go there, find the neighbors, with the highest similarity score using the embeddings, retrieve all records of relationships, get all the relations, and also get all of the like text data. And then within the context window you have, allocate some space for each of the things you found. So you found 10 results that seem like useful for this John node. You put them all in a context window and then move on. And then give that to LLM to finally generate an answer. So to summarize, in a graph, you have a question, find the most relevant node, explore the neighbors, gain the information from neighbors and the node itself, give all of that, fit all of that text into a context window, and then give it to LLM to answer the question. That's it. That's local search. And then the most interesting one is global search. So global search, like I said, is best suited for questions that can only be answered with comprehensive understanding of the data set. Now, the way it works, like I said, is you have community reports. Like I said, every community has a community report and it has a community summary. Summary is just like a short version of the report. And you gather and fit all of the community reports into a context window. So you know what is a useful and non-useful community report. Based on that, you take all the useful ones, put it into a context window. And for each community report you have gathered, you will make an LLM call. You make 8 LLM calls if need be. You make all those LLM calls, you get all those answers, you give them all a score. When you're in the prompt, you say score the thing. So you get all the scores, you re-rank all of the scores based on like best to bad, and then remove all of the answers that have score less than a particular threshold, and then keep the ones and then fit the rest of them into a context window, and then send that to an LLM for a final full comprehensive answer. So that's global search. Cool. So GraphRAG, there are some limitations. No one likes to talk about it, but there are some limitations. Cool. So what are those? I mean, I'm saying no one wants to talk about it because all the Microsoft internal presentations I've seen, no one talks about the limitations. So this is I think a good slide for you. So latency is a major thing. So local search is fine because it's pretty similar to what vector search, normal naive RAG vector search is. However, for global, it is going to take long time, sometimes 20, 30, 35 seconds. But the good thing is LLMs are getting faster, so that's one good thing. Anyway, so latency for global search depends on many things. One of the key things, is the community level. So if you go to the bottommost community, you have so many nodes to take care of. It's so much computation. If you go to the top, it's too less information. Like it's quick, but it's too less information. So you put it in the middle, basically. You have 4 community levels, you take the middle one. So it took me $3 to build 5,000 lines with GPT-4 Mini. Still expensive, I think. For the interest of time, I'm going to skip over to the demo. I think I need to yeah, cool. I'm going to show you all the demo. I think hopefully this is interesting. Show the window. Cool. So you see this is the graph. This is the knowledge graph generated. If I zoom into it, let's go there. You can see all the green things here. This is by the way the data of BBC News over the last 3 months. So this is the knowledge graph for BBC News over the last 3 months.
How did you generate it?
I indexed it. I mentioned indexing process. So you run a command saying literally python-graphrag.index equal to take this data, and then it will start indexing, and then it will eventually give you a .graphml file which you can use to visualize stuff. Cool. So the one interesting thing I'm going to talk about here is this. So if I zoom in here, all the green things are attacks and stuff. if I go to random color, all these colors are basically looking like, it looks like Olympics and Paralympics, doesn't it? Can you see them? Yeah. Can you see them? And if you go to the yellow, you can see Harris campaign, all this like politics stuff. Yeah. So this is not important. This is not useful, is it? This is not useful. So for the final visualization, I'm going to show you this. So that's the graphic. So you can ask any question you want, and it will use this graph. So let me just quickly show you how the data is stored in the actual knowledge graph. Let's just say, like, just tell me a name. Any name? John. Okay, John Murray. Okay, John Murray. So if you click that, you can actually see who John is. As BBC sport commentator, I didn't know that. And you can link nodes are England, and the link relationships are like essentially like you can see some of the relations here. So that's one example, right? So just give me another one. Oh, nice one. Lewis. Hopefully he's in there. Has there been news about Lewis Hamilton in the last 3 months?
LOUI.
No.
Oh, sorry.
Oh my God. Okay.
My bad.
Okay. Just give me some random name, random object.
Kesteren.
Oh yeah, go for it. Let's go for it. There you go.
All over the news.
Okay. Oh man, that's crowded.
Told
So
you.
you can see how many relations someone has. So do I have time for making a query?
Yes, please.
Okay.
I think everyone wants to see
So we
it.
can actually ask about what he is doing and his relations and stuff. Not personal, of course. It's gonna get crowded. The reason you see all of this is basically. So yeah. Let me just ensure my backend is running. So we will like. Yeah.
Obviously, this is a snapshot in time of the last 3 months. So
I know what exactly you're gonna ask. That's a
So-
brilliant question. Yeah.
So obviously, this is an evolving model
Yeah.
where the relationships are going to change and such. So in terms of the usefulness of it, does it take an expert to recognize an expert, or are we only seeing just an evolving relationship in all of these things?
So one consideration I had in my slide was when you make this graph initially, you index it, it's impossible almost to actually evolve that. Because you take the summaries to the top level, right? You make so many assumptions that everything below is complete. You make summaries based on that. So if you want to ingest some information to the bottom entities, you have to change the entire thing. And adjusting that is not supported. So you have to re-index the entire thing, which is expensive. So yeah, I guess hope that answers your question.
What if you started this whole process through some temporal nodes and identify relationships which have dynamic temporal nature versus the ones that do not have? So temporal graph is also a research area.
Exactly.
Makes sense.
So that is something not supported by GraphRAG, yeah. However, I think there were some talks about indexing indexing based on communities. So like instead of changing from the bottom, you change from the top. So change the community, change the community summaries, change the entities, eventually like inject in that information from top to bottom. So that's one being internally explored, but it's not released yet. But that's a good question. Yeah. Thanks for that.
Sorry, just another thing. This is really like a zeitgeist. So whatever is popular at a particular point in time, talked about, is what we're seeing here in terms of great for media.
Yeah.
And whatever bandwagon we want to jump on, the AI bandwagon.
Yeah.
This is sort of great to hear.
Yeah. Yeah. Is that a question? Sorry.
No,
Okay,
it's
cool.
just me.
Yeah, I mean, that's a good point. So like knowledge graph has been around for a long time. Everyone uses them. Google uses them. Everyone uses them. It's been a proven technique. So what we're seeing is AI with it. AI is known to hallucinate a lot. If you can restrict that hallucination using knowledge graphs, I think it's a great thing to be looking at. And it doesn't seem like I I don't know how like bandwagon it sounds, but I definitely think it's scientific and supported by graph theory. And blockchain, unlike blockchain, maybe AI is completely different and it's proven by like the relations and stuff.
So do you go to the foundational levels of knowledge graph, because that talks about entities, relationships, but also object properties and data properties. So do you go down to that level of detail as well when you formalize these relationships?
So when you make the relationships, all you do is there is some metadata there. So there is a metadata between relations. That's a bit of two level to talk here, but there are like weights on each relationships, and then there are like embeddings of like other nodes. That's like all the metadata. But I don't know if there's any more deeper, anything deeper than that. I think you can't see in the data as well. So if you just like look at this diplomatic primary, you can see all the data there is about this entity. So there is nothing more essentially.
I was just thinking about how do you reduce the computational complexity of reindexing every time. So if the knowledge graph has data properties and object properties, then all we need to do is to refresh that. I'm thinking about a weather example. So temperatures change, but the relationship does not change.
I see what you mean.
The entities and the relationships. So maybe we do not need the reindexing. We just need the refresh of these data properties and object properties.
I definitely see where you're going. I think that is definitely possible, but I'm not sure if it's possible with this current iteration of what GraphRAG we have. But I can see it being a possibility if all the magnitude data, all the number data is one place, agnostic of like all the relations. If that's the case, we can it's a matter of removing that node, adding a new node, right? But if it has relations, then it's kind of hard.
Sorry. In terms of the weighting of the graph node, if we just imagine each of us as a node with some knowledge, I know there's one expert here who knows a lot about proteins. How do I weight all of that noisy data? And is there a way to automate that? Or is there modeling within graph data to help filter out those relationships?
Yeah, exactly. So that is exactly what somebody is doing essentially. So let's say you're an AI expert. Let's say that person is the blockchain expert, right? You have something about some X thing, and they have also said about some X thing. So the X thing is your common thing, right? So X thing is the common theme you have. So when you ask question saying, tell me about the people who talked about AI and blockchain about X. The summaries that are above you, the people in front of you know what you have, right? So they will say, this person has this. And you from that, another summary, that will say, this person also talked about X. So at the top node, you know for a fact that community 1, community 2 are actually talking about one thing. So when you give the LLM, you give your entire thing, your entire input, and your entire input, and then LLM will take care of inferencing the actual information essentially. Can I
finish your query, please?
Yeah, so many questions have been blocking me from running this query.
Let's finish this query and what he's demoing, and then I would like if you have more questions one by one, so everyone.
Okay, cool. I'm going to run the query. No questions for now. Okay, where were we? My terminal is running, right? Let's stop this. Oh, thank God. Okay, cool. So we run the API. This is an API. It's a repository. I can all share in the later. So what it does is, if you go in the settings.ml, YAML, it exposes BBC News. This one. So that is the thing we have. So now we need to essentially upload this stuff into the front end. So this is a backend for the web app you saw. Cool. All we need to do is refresh. Upload from the BBC News. Come on. Recording. GraphRAG. GraphRAG. Where's API? Sorry. Yeah. Artifacts. BBC News. There you go. Cool. Now we just load all the Spark files and hopefully you can chill. Yeah, okay, cool. So now if I include the communities and text and search, cool. It's not running. Of course, messed up. My port is running in the background. I'll get a temporary fix now. Where's API? Can't change it. Does anybody know how to remove a port from unbind a port? It's fine. I just use my.
Maybe just use another port.
I can't change the ports from this. Sorry. You know what? I'm gonna be like AI person and then go all AI on it. Let me show you something. I think you know all what I'm talking about. Well, WAP. Everybody knows WAP. Unbind. 8,000 was it?
88 wasn't
88, yeah. 88, 8,000. 8,000 port. Jesus, sorry. Oh, that's the 1 second. Let me just click that. There we go. Oh, thank God. I just done it. There you go. Fine. Okay, cool. Does it work? Will it work? It works. There you go. AI. I told you, AI is not not blockchain. Let's refresh. Ask. Okay. We are ready for a question about last 3 months BBC News data. What are we talking about? Kesteren, is it? So what do I want to ask about Kesteren? What is he up to, you know? I'm not gonna get political here. Like, I'm not gonna get like political here. Okay? The correct spelling? EI. EI? Yeah, of course. What is Sama been up to? So, is that a local question or is that a global question? No, it's a local question because you're one entity, right?
No.
It's one entity. Kesteren was one entity, and then all the relations are used.
Very broad.
I have one question about global search. Okay, beautiful. It understands the holistic picture very well.
Yeah.
But when you are really looking for something like very text match, exact text match, when you want to, for example, search a company around all.
Okay, name a company. Come on, let's search it.
Yeah, sorry.
So, can you give me an example? Like, you want to like run the global search on?
So I can say that.
Okay,
Well,
which political
we say that
leaders
it will be a
have
local
here?
search when you do such an operation in that case. That company will become local, and then you will do around that. Okay. So rethinking about it. Imagine you use the global search function, and you will be using it for more general things then.
Yeah.
Only more. Just give me the general idea about how this knowledge base look like.
Yeah. So someone said one question. I'm gonna show you one question. I think that will hopefully answer your question. So I'll take a good global question. Okay. So most odd or unusual news stories. How can GraphRAG even answer that? There is no base, you know. Like, so let's ask that. Okay. So if I ask that in a global search, it's gonna take a while. So most odd news, how would you even like know what's odd? You know, or how do you do compute vector search, vector embeddings for odd? You know, unless this is odd news, something happened, but you can't know that. Okay, there you go. So odd news.
It's sad news. I don't know if it's.
It is sad news. I think I know exactly why that is. You know why? Because this is trash. GPT-4o mini is not really good, despite what popular opinion says. So let's just see what it says now. Hopefully that doesn't get cached. I think it will get cached. It did get cached. So what I asked right now, since I'm asking again, that answer and question has been cached. Oh, should have been better phrasing there, but hopefully. Several reasons, unusual and tragic nature. So murder, and knife attack, children in Southport, that is fair enough.
They sent from Disney or something.
Yes. So actually, I have a question on that. What if I try? Bear with me 1 second, right? What if I try this question? List all legal lawsuits in the data, right? So listing information, comprehensive information, that is what I mean, like, like trying to understand the entire data. You wouldn't know this until you actually gone through the entire data, right? Let's see what happens. So you can also see it took 8 LLM calls. And that's not milliseconds, by the way, seconds. All right, cool. That's one good, because we saw Katie Price there, so that means we know it's a lawsuit. Axel, Ricky Jones charges, Hugh Edwards, Ashley. I mean, you can see all of these are pretty. It's a comprehensive list. It is a comprehensive list. It's not like 2 or 3 examples. And you can also see the actual data it gives you. So there you go. And not only that, you can also see the notes related to this. So this is our communities. So that means there is no node here because it's a comprehensive information, right? If you do local search, you can see the community entities. But since it's a global search, you use community summaries to answer your question. Hence, there is no individual entities there. Yeah, that's
on local search. How do you recognize the entity that you start looking for nodes?
Yeah. So as I mentioned, when you do local search, so what's your name, sorry?
Ivan.
So let's say you released a new phone, right? What did Ivan do? And then it uses Ivan, finds Ivan in the news data set. One of the news says Ivan went to America. Ivan went and released a new phone. So these 2 are captured and then it will return. So the entities are Ivan. All the relations to Ivan, all the things happened around Ivan are just beside him, like just beside Ivan as a relationship to another nodes. So Ivan, America, has gone to. Ivan released phone. So like it's all around the actual centric object. So just grabs that.
Okay, but what if you have 2 objects? What if it's like Ivan stole John's phone?
It doesn't matter as long as it's just naive RAG, right? It's just basically naive RAG, but considers its neighbors essentially. That's yeah.
I'm going to step in. Let's say 4 more questions. I see a few hands waiting to ask. So do you have anything else to kind of say, and then we will just go to questions?
When you hadn't finished
I know just that because.
Okay.
So do you want me to finish it off and then go there and ask questions? Or
Yes, please finish off a few minutes.
Yeah, absolutely. So that's it, folks. GraphRAG. Hopefully you enjoyed it, and please feel free to reach out to me. If you have any other questions.
What happened to the Kesteren query?
Sure. That was
Actually, I'll come over there. I'll show you. Okay. So that's my show sense. If everyone's anyone's interested.
So a few questions because I just see the hands. I've seen you.
I'd like to challenge you on the known unknowns.
Okay.
So there was this Mike Ashley guy who died recently off the coast of Sicily. I know he won a lawsuit because he was the guy of alchemy fame. Why was he not on that list?
It's because context window. So my settings says I am accepting 4K context window. If I increase that, and if I tell my model to list specific number, it will try and get that. So also, what was the name of the person?
I think it's Mike Ashley.
You know what? Why don't we look him up? What's the name?
I think it's Mike Ashley. Alchemy was his company. Mike Lynch.
Mike Lynch, the billionaire, of course. Yeah, the billionaire. Okay, let's see what happened to Mike. I mean, I know he was passed away there. So if I just focus on US fraud trail. If you ask about Mike Lynch, if you ask about any fraud trails, you probably pick it up. It's British, it's tycoon. Oh,
But he won his lawsuit.
he won his lawsuit.
Yeah, it's he was
Yeah, I mean, like all I'm saying is, if we make it say, find me 15 ones, it will try and extract more. All it does is like extraction, right? Yeah.
Next question. You had a question there with the, sorry, you in the glass, you had a question next to you.
Oh, yeah, so I was wondering about which part is the most token consuming technology?
Token what, sorry?
The token consuming. Which one?
For me,
Or something.
which process uses most tokens? Indexing and global search. So global indexing makes sense. You send all the data into the AI and then extract it. So in global search, also similar. It's like it depends on the number of communities you're looking at. Like I said before, there's the levels. So I'll quickly show you that. So if you look at the data, these are communities, yeah. So community has level 000, and so on. So I'm up to level 4. Level 4 is the topmost. So hence there are less number of level 4s. So level 4 only that. So if you set your level number when you query to high, it will be like so low. So it depends completely on what level you're setting up. Usually it is recommended to keep it in the middle, so like 4. Like 2 is probably the best one because you have balance between the number of computation you have to do and latency, like how much information that there is. Yeah. Yeah.
Can you say tokens by doing summaries on just the subsets of what communities or some later?
Then what if there's a question on something that is missed out in the summary? So you're saying, so what you're saying is, why don't you summarize only some important like entities, right? But what if there's a question on the unimportant entity, right? So you need to summarize everything, but maybe when you use, you're not going to look at the unimportant stuff anyway, because you're going to reduce your search space, right?
Yeah, so why do we summarize on those communities?
Sorry?
I think the summaries, they are multi-layered, so they are physically different level of abstraction of the same thing. Like, can you?
I see what you mean. Okay, because it's like a tree, and okay, I see what you mean. Because you have some nodes here, you have summary, and then the summary above it will also have this. But you have to consider the fact that the summary above it, the community above it, will also have other things around it. So it's a mixture of it. So it's basically pointing, locations. It is. I see what you mean. It is kind of hard to achieve that. Yeah.
Yeah, I'm just wondering about like reducing.
I'm just going to step in again. So just thank you very much. Thank you.
I got to thank you all, like for letting me talk, letting me bear me for like such a long time. But, hopefully that was useful.
And just once again, I want to say thank you as well. I didn't mention in the beginning. There's a team from Nexgen Cloud as well who were working behind, and just they were mentioned. So just want to say thanks for them as well. Thank you very much.
Thank you very much.